Good afternoon, everyone. So I want to thank first Professor Debashish Chaudhary and Indian Academy of Sciences to give me the opportunity to give this talk. So the title of my talk today is to understanding principles of design of molecular machines, and I'll be giving a structure biology perspective to this symposium. So in terms of looking at molecular machines, so many works, like mainly the biophysical studies, in especially molecular imaging of the cell, have shown that the cell is composed of elegant, highly organized molecular protein complexes, and these protein complexes perform all the cellular function in the cell. So the term molecular machines was mainly like it became widespread after Bruce Alberts in his view in 1998. So he was the first one to use the term molecular machines for protein complexes which work inside the cell. And the reason why he termed it as machines was mainly that these these protein complexes, they contain highly coordinated moving parts. And this has been very well introduced by the previous speakers, Professor Debashish Chaudhary and Dr. Roop Malik. I would like to show you an animation of made by Harvard Biovisions. It's on YouTube, and this very well shows, illustrates the different molecular machineries which are present inside the cell. And some of the machineries which were talked by the previous speakers can be seen here. And so in the cell you can find different molecular machineries which are there. So here is a proteasome which going through, and this is the mitochondrial membrane. And what you see here are the ATP making machinery, which is using the proton efflux to make the ATP inside in the mitochondria. And so as you go along, so you see the rotatory function of mechanism of the ATP synthase, the nuclear pore, which helps in the transport of materials in and out of the nucleus, the ribosome, the center of the ribosome, where additional single amino acids are being added based on the polypeptide, the mRNA codon, the flagella, you see the rotatory movement of the flagellum, showing which helps the bacteria to move. So you will see different molecular machines inside the cell doing their function. So what you eventually see in all these molecular assemblies, that their movement are highly coordinated. So this is the microtubule that the previous speaker was talking about, and the motor protein walking along the microtubule with the load there. So what we see in terms of in case of biological machines, which are actually macromolecular machines, that they are complex and they are composed of multiple component systems. So like for example here the ATP synthase, which makes the energy inside the cell, it is formed of multiple polypeptides, and each polypeptides are coded in different colors. So the ribosome here, again, it is made up of multiple proteins. So it is essentially two subunits, which is not clearly shown here, but it is made up of two different subunits, the small subunit and the large subunit, having multiple proteins in it, and three RNA molecules, and spliceosomes, which are again a large molecular machinery which helps to remove the introns from the mRNA. Now, what the other essential features of these machines are that they operate under executive control. That generating a motion in the molecular machine is not important. What is important is to have the control on these machines, and so these machines work in a controlled fashion, and they are directional. And mostly we see a translational as well as rotational movement in these machines, and these movements are with precision. So you see rotational movement in this ATP synthase, whereas you see a ratchet-like movement between the two subunits in the ribosome. So I'll come to the ribosome in a little bit. So what we see that the macromolecular machines essentially have these features, and they also operate at a very high speed. Now, it is very essential to study these machines from a biological context, and also from the context of chemist, as it provides a strong incentive to design nanostructures. So this is a statement from a review written by Bernard Faringa, who shared the Nobel Prize last year for chemistry for the development of molecular machines, and he states, understanding and harnessing such phenomenal biological machines provides a strong incentive to design active nanostructures that can operate as molecular machines. And for the molecular machines, he goes on further say, it is conceivable that molecular motors and machines will play a prominent role in the nanotechnological revolution of the 21st century, as their macroscopic counterpart, the steam and the internal combustion engines, played in catalyzing the industrial revolution of the 19th century. So it is essential for us to understand, not only for the context of the biology of these macromolecules, but also to provide a strong incentive for the chemists there. So in terms of structural biology of the molecular machines, there are primarily two major methods by which the structures have been determined of the molecular machine. Mainly X-ray crystallography has been the choice of structure determination till now. In X-ray crystallography, the protein or the macromolecular complex is first crystallized into a three-dimensional, and then this crystals are then, an X-ray diffraction data is collected out of this crystal. So where you shoot the crystals in an X-ray beam, and what you compute out of the X-ray diffraction pattern is an electron density map, and then you fit in the model to generate the three-dimensional structure of the macromolecular complex. However, in recent days or recent years, cryo-electron microscopy, which is another technique to determine three-dimensional structures, has been so the technical advancement in cryo-EM has enabled or has made this as a more like this is being now routinely used to determine the high-resolution structures of the macromolecular complexes. And the advantage of using cryo-EM is that it removes the bottleneck of crystallizing the sample. So the sample can be layered on an EM grid, a thin layer, a small amount of the sample, and plunged frozen in liquid ethane, and can be viewed in the microscope. So these small dots are actually one molecule of the macromolecular complex. Multiple images are collected, and we pick up the particles. So each particle is picked up. They are then combined, like similar views are combined in 2D class averaging, which are then, or the 2D class averages are then combined to give the three-dimensional model of the macromolecule or the molecular machine here. So coming to the machine that I'll be talking, or I'll be using as a model to talk about molecular machine, the ribosomes. Now, ribosomes are the molecular machine which reads the genetic code, which is the nucleotide code in the mRNA or the template, and it forms a cognate polypeptide. So it reads the codon in the nucleotide codon in the mRNA to form a sequence of amino acid based on the nucleotide, based on the codons there. So in order to do this function, it has to do three different jobs at the same time. One is to decode the codon and so that it obtains the cognate sequence of amino acid. Secondly, it has to form the peptide bonds between the amino acid, and third, it has to move along the mRNA from the start to the stop codon. Now, ribosomes have divided this three functions to three different sites, or they use a decoding device, which is essentially a site in the small subunit of the ribosome, where they decode the codon in the mRNA to obtain the cognate amino acid there. The peptide bond between the amino acid is made in the peptidyl transfer center, which is it's essentially an enzymatic activity of the large subunit. And in order to move along the mRNA, the two subunits come together, and the ratcheted movement, the movement of the ribosome with respect to each other, help in the movement of the ribosomes along the mRNA path. So in terms of biology, so this process of translation is divided into three steps: initiation, elongation, and termination. This was briefly touched upon by Professor Chaudhary. And in the first step, that is the initial steps of translation, the small subunit, it binds at the 5' end of the mRNA, which or the start codon in the mRNA template, and this specialized tRNA, which is an adapter molecule which helps in decoding the codon with the cognate amino acid, is recruited to at the start codon, and followed by the assembly of the whole ribosome, that is the joining of the large subunit to form an elongation competent ribosome complex. Now, so this ribosome is now competent to move forward and decode the remaining codons in the mRNA till it reaches the stop codon. So the next step is the elongation step, where the ribosomes decode individual codons as it moves along, and thereby the length of the polypeptide increases. And when it reaches the stop codon, then it no longer has an adapter molecule for this. So this is now recognized by release factors, which then release the polypeptide, and the ribosomes subunit then are recycled. So this is an oversimplification of the process of translation that happens in 3 steps. And so I'll show you an animation of the working of ribosomes from Venki Ramakrishnan's lab, which essentially shows in an animated fashion how this whole process happens. So initially you have the small subunit, the mRNA template, and the initiation factors, which are then assembled on the ribosome. So you see here the mRNA and the initiation factors with the tRNA. This is the initiated tRNA which will be placed on the ribosome. The mRNA binds in the mRNA cleft, and this is the formation of the initiation complex, then recruits the large subunit. The formation of the whole ribosome then leads to the removal of some of these factors. They are then released. And next step is the elongation step. So initiated tRNA is there. So the next tRNA is then brought about by another protein factor called EFTU in bacteria. It recognizes, now decodes the next codon. With the GTP activity of EFTU, the tRNA will flip inside towards the peptidyl transferase activity. So it will transfer them in. So the peptide bond between the 2 amino acids happen here. And so now the next step is to move along the mRNA path. So this movement is brought about by the movement of the 2 subunits with respect to each other, and by the energy provided by the GTPase action of another protein factor called EFG. So this helps the tRNA, the ribosome to move in steps along the mRNA path. So here you see another next tRNA which is now being decoded here, and so this process of elongation is then repeated throughout the length of the mRNA path. So, and this happens at a very fast rate of in terms of milliseconds each amino acid is attached. And you can see here the polypeptide chain which is coming out of the mRNA tunnel. Now as it comes out, it can fold inherently by itself or by the help of another protein factors or chaperones. So this happens till you see a stop codon, so which is then recognized by release factors, and what it does, it releases the polypeptide chain, and then the release factors, ribosome recycling factors are then involved, which will then break open the ribosome complex to recycle it. So this in short tells how the ribosome functions. Now in terms of to understand the principles of design of molecular machines, I'll just focus on the initial steps of initiation, and we'll try to understand how looking at the initiation complexes, if we can derive some principles for the design of molecular machines. Now the initiation, as I have mentioned, it is the process where the where the ribosome recognizes the start codon and places a specialized initiator tRNA at the start codon. Now, this process of initial steps of translation is extremely different in the two systems, that is the prokaryotic, which is the bacterial system, and the eukaryotic, which are the higher organisms starting from fungus to humans. So, so without going into the details, what I would like you to focus is that in bacterial system, there are three initiation factors or polypeptide helpers which help the ribosome to place the correct tRNA or which is the initiated tRNA and recognize the correct start codon. So there are only three members and which are essentially three polypeptide chains. Whereas in a high system, it goes through multiple steps, and there are at least 8 initiation factors are involved, and some of these initiation factors are multi-protein complexes themselves. So range from 18 to 25 polypeptides are involved, or they help the ribosome to locate the correct or the cognate start codon and place the correct initiated tRNA at the start codon. Now, if we look at the overall structure of the ribosome or the small subunit, so this is how the structure of the small subunit looks like. Here I have shown the small subunit from eukaryotic system, which is the 40S, but the structure of the small subunit from the bacterial system is essentially the same. So the overall structure is the same, and this structure will roughly remind you of a bird, and this is essentially how this the parts of the ribosomes have been named. So this forms the head of the small subunit with the beak region here. This whole forms the body of it of the small subunit, with this region being called the shoulder. This forms the platform, and the two left and right spurs, which are the left and right foot. And these two, head and the body, they are attached by, which is not very clear here, but it is attached by a helical RNA helix. So there is only one copy of rRNA here, which is shown in different colors based on their different location in the body and head. So the neck is formed by an RNA helix that I'll show you later in the presentation, and what you can see is a cleft or a channel in between the body and the on the head, which is where the template for translation or the mRNA sits. Now, this 40S is actually composed of multiple components. So here all the 33 all the 33 polypeptide chains are shown, so in the individual structures. Now, what we have to appreciate is that all these different polypeptide molecules, they come together along with one rRNA moiety, which is about 1800 nucleotides long, to form this subunit. So here to show you that this is the in gray is the RNA molecule, which forms the core of the ribosome, and the different polypeptides or the proteins, ribosomal proteins, are attached by binding to specific regions in the rRNA chain. And this is the front and the back view. So here is the beak, head, and the body, and in this case the back view, so the beak is here. So what the structure of just the 40S or the small subunit tells us is how the individual components are arranged. So just looking at this particular structure, what it reveals is the overall design of this particular machine or the macromolecular complex. It shows us how these multiple components which it is composed of, they come together to form it. We can get an idea of which are the rigid regions in the machine, in this macromolecular complex, which are the flexible regions, the interface between the subunits, what are the accessible regions, what are the accessible surfaces in the macromolecular complex. What it can also give us is some insight into how this machine would work, together with additional information and from other studies, and these informations could be coming from other biological experiments or mutational, like something like biochemistry of the system, or by mutational data or the genetics data. So However, in structural biology, what we do is we try to capture the molecular machine in action, which by which I mean that we determine the structures of these complexes in different states. And if we do that, so this is the I'll just present briefly the work that I did in my postdoc in Venki's lab. So we captured different multiple the initiation complex from both bacteria and higher organisms in multiple states. And what in terms of what we learned from that is that the ribosome is actually present in multiple conformations when it goes throughout the initiation pathway. And you can see here that the lines, the red, yellow and the purple, they do not overlap completely, so they are in different conformations. What we also learned is that binding of these helper proteins on initiation factor, so these can either bind to a given conformation of the ribosome, or the binding of these factors can shift the equilibrium to one of the conformation. So for example, in this case, if these two factors bind, we always find the head in one fixed conformation. Otherwise, in the absence of it, it can wobble, or it can rotate and close and open. Now binding of these factors, they change they cause conformational change in the P site, which is the site where the tRNA binds, or the initiator tRNA binds. So this is essential for the recruitment of initiator tRNA, and the initiator tRNA actually binds or it prefers an open conformation of the ribosome. So by open conformation I mean that when the head of the ribosome is further away from the body. So in yellow is the open conformation of the head, whereas the body remains the same. So the open conformation of the head, and in the blue is the closed conformation. So and what the other two conclusions that we drawn was the closed conformation is actually essential to scrutinize the correct the selectivity of the correct tRNA and the codon. And we all, apart from the multiple conformations of the ribosome, we also saw the multiple conformations of the helper proteins or the initiation factors themselves on the ribosome, and this may be essential for their eventual release from the complexes. Now, in terms of, I'll just quickly show the movement that we observed. So the ribosomes undergo two type of movement. One is the rotation of the head with respect to the body, and you can see the neck region of the ribosome, which is a helical region here, and the other is the opening and closing of the ribosomes. If I can open this, okay. I'll show this, the open conformation. So this is now a ribosome in an open conformation, and now it closes down. So and this is the neck region, and you can see it is relaxed here, and when it closes down, it compresses. So this is the ribosome conformation that we observed in the eukaryotic system. In the prokaryotic system, we saw exactly the same thing, although the number of factors which are involved are different, the number of proteins involved, the numbers as well as the polypeptides are different, but the ribosomes, they undergo the same conformational change, rotation as well as opening and closing of the ribosomal head. So in this view, you can see this is the ribosome rotation, which is causing the P site, which is here where the tRNA binds. It causes a change in the tRNA, the P site region, the opening and the closing. So you can imagine open conformation will be more helpful for the tRNA to bind there. So, and again, so here you see an mRNA latch, which is a kind of a door to allow the mRNA to come in and out. So the different head positions allow the mRNA latch to open and close. And this is the neck region, which is highlighted here, the helix 28. You can see it undergoes compression and relaxation when the ribosome undergoes these movements. So it's a spring action of the neck region that you see. And this movement of the movement of the ribosomes, especially mainly the head movement of the ribosome with respect to the body, that essentially drives all the biology of the initiation complex of the initiation. The tRNA bind in an open conformation, and when the head swells or rotates, it brings the tRNA closer to the body so that it can be it can be monitored and looked into whether the tRNA bound is the correct one or the non-cognate one. If it is a non-cognate, it is removed. So essentially, looking at all these molecular complexes that we have learned from initiation, what we can infer about the design of molecular machines. So what we have seen here is that a loosely associated bulky head, so the ribosome had a huge head on a body, so a bulky head of the small ribosomal subunit, which is attached by a narrow spring-like neck to the body, and this allows multiple conformations of the ribosome. So if we leave it alone, the ribosomes would keep achieving different conformations by itself, and hence the motion in the ribosome in the small subunit. So it looks like, although we, based on our complexes, it looks like that the Brownian motion that the complex sees, it is sufficient for its rotation of the head as well as opening and closing, because in all complexes we didn't use any energy source. So it is sufficient, and what we feel is that the binding of these initiation complex, these initiation factors, they may provide a directionality to the motion, because we saw that once we have binding of certain factors, then we see only certain conformations of the ribosome which is favored. So so here the binding of ligands could be used to provide directionality. Energies from GTP can also be used, but in this case it was mainly the binding of ligands that we had seen. And what we also see that there is a lot of flexibility in the molecular machine, or the ribosome subunit that we see here, and a lot of intramolecular movement, and these are really essential because it provides different conditions of attachment and detachment of large subunits. So large ligands. These ligands, like the tRNA, the different initiation factors, they are huge ligands which are attached to the ribosome by non-covalent linkages. And by having a lot of flexibility and intramolecular or different conformations that they scan through, what it essentially does, it will have in certain conformation partially formed contacts, and in another it will have partially detached contacts. So it can, so the binding and dissociation can proceed via many intermediates, and Professor Chaudhary was referring to these intermediates in his talk there. So you can have the binding and the detachment of the ligands via multiple intermediates, and these intermediates will have partially formed or partially detached binding contacts. And essentially even the binding, as I have mentioned earlier, the binding of the ligands can provide directionality to the system. So with this, I would like to thank, so the work that I just showed on the initiation complexes were done in my collaboration, in my postdoc work in Venki's lab. So I would like to thank him and his lab members and the collaborators at NIH and ISC, where I'm based now, and Indian Academy of Sciences for giving me the opportunity for this talk. Thank you.
Hello, here. Is there a mobility in L7 and L12 stock region of the large subunit?
Yes, there is a movement, but since I was talking mainly about the, but L1, the central protuberance, L7, L12, they also move. So
all the machinery required for translation seems to be present in the small subunit.
Yes.
So what role does the large subunit actually play?
Okay, so when I said that there are three major functions or three different jobs, one is the decoding, which is done in the small subunit, but then there is a function of making the peptide bonds. So this peptide bond function is in the large subunit, and then the movement of the ribosome along the mRNA channel. So in the elongation step, it is actually the movement, the rotation between the large and the small subunit which drives it, and apart from the the energy cost from the EFG. Sorry.
Okay, so let's thank the speaker once again for a nice introduction to it.